Okay, so welcome to today's session. We are going to continue looking at epithelial tissue like we started last time. So today we are going to end where we're going to start where we ended last time, and let us do a quick recap of last session. Last session we did say that the human being is made up of four basic tissue types. The first basic tissue type that the human body is made up of is nerve tissue. This is the one that is specialized in transmission of nerve impulses. Then we have epithelial tissue, which forms glandular tissue as well as specialized for lining body cavities as well as covering body surfaces or organs. The third tissue type that we looked at was that we just introduced is muscle tissue, the one which is specialized for muscle contraction, the one that is specialized for movement. Okay, so that is muscle tissue. And then the fourth one will be connective tissue. And connective tissue, we said that that is the tissue that is specialized in support of all organisms. And we did say that every organ in the body is made up of one, the parenchyma, and two, the stroma. The stroma means that supportive tissue, the supportive tissue that does not perform the actual functions of that particular organ, but is only there to support. All right. So, for example, if you're talking about, for example, the kidneys, or maybe let's say the liver, you have in the liver, you have cells known as hepatocytes, which are the ones that actually carry out the functions of the liver. They're the ones that carry out detoxification. They are the ones that carry out the formation of bile, okay? But just around the hepatocytes, you're going to find certain structures that are just connective tissue, just there to support the actual parenchyma, and that is what is called the stroma of an organ. The stroma of an organ, stroma, that is the stroma of an organ. So in epithelial tissue, we looked at epithelial tissue last time. We talked about the main characteristics of epithelial tissue. We said that the cells are closely opposed. We said that they exhibit polarity. We talked about the nucleus, the shape of the nucleus being in relation to the type of cells, in relation to the cells of the of that particular epithelial tissue. For example, columnar cells will have ovoid ovoid shapes or shaped nuclei that are elongated. When you look at squamous epithelium, you're going to have flattened nuclei. When you look at cuboidal epithelium, you're going to have spherical or round nuclei. And when you look at the positioning of this nuclei, in cuboidal epithelium, you're going to find that the nuclei will be centrally located. It will be right in the middle of the cell. But when you look at columnar epithelium, the nuclei will be towards the base, near the basement membrane. And so that is one way you're going to be managing to identify what epithelial tissue you're looking at by just seeing where the nucleus is and the shape of the nucleus. It will be able to show you. We also talked about other types of epithelial tissue, which are not sheets, but they're aggregated together. They may not have a basement membrane, but they are epithelial cells and they carry out functions of epithelial tissue. We talked about the shapes of the epithelial tissue, squamous, cuboidal, columnar. Okay, we went ahead and talked about the special classifications, which is pseudostratified and transitional. We then went ahead and talked about the modifications, on top, the apical modifications of these cells. okay, where we looked at the cilia, we looked at villi, we looked at cilia, we looked at villi, we looked at stereocilia, we looked at okay. So these apical cell specializations or cell modifications, this is one of the characteristics of epithelial tissue, where you're going to find different things on the apical domain of the cell. So for example, some cells are going to have microvilli, okay? Microvilli are tiny projections on top of the cell. These help in increasing the surface area of a particular organ. And microvilli is especially found in the small intestines, and it increases the surface area for absorption of nutrients. So when you look at the intestines, you are likely to see what is known as a brush border. When you look at the intestines under a microscope, you're going to get this intestine and you're going to cut a small portion, and then you look at it under a microscope. And then when you look at it under a microscope, it will look like a brush, like a shoe brush. Remember, when you're looking at a shoe brush, you're going to see that on top there are those tiny projections on top, and therefore that is what the brush border of the small intestines will look at, the ones that has microvilli on top. So microvilli are different from villi. They are two different things. When you are talking about a villi, you're talking about, first of all, the small intestines are highly folded. They are highly folded, but within those folds, you find finger-like projections. And those finger-like projections are the ones that are going to be known as the villi. And then on top of the villi, you have a layer of epithelial tissue, of simple columnar epithelial tissue. And this simple columnar epithelial tissue has tiny projections on the apical surface of them. And therefore, those tiny projections are the ones that are known as the villi. Okay, so those are the ones that are known as microvilli. So finger-like cytoplasmic projections on the apical surface of most epithelial cells. So those are known as those are the ones that are known as microvilli. And then you have stereocilia. Stereocilia is also known as stereovilli. The reason is because it resembles microvilli in the structural makeup. The internal structural makeup is like the microvilli. So what you're going to find, where you're going to find these stereocilia would be in organs of absorption also, all right, as well as, so some of those organs include the epididymis. So this particular portion of this structure is a part of the epididymis that was chipped off, and then it was seen under a microscope. This is the same one here. The only difference is the staining that was used. So here it's appearing pink and here it's appearing purple because of the stain that was used. Now what you're going to notice is this epididymis will have these things projecting on top, and these are the stereocilia. These are the stereocilia, and therefore you will notice that there is also a brush border appearance of the epididymis. Now, because the only difference between stereocilia and microvilli is that the stereocilia will appear very long, much longer than the microvilli, and so that is one major difference. The other surface modification would be the presence of cilia. On top of some cells, you're going to find hair-like projections on top of some cells. You may be able to know some of these areas such as the respiratory tract, the air passages of the respiratory tract. These are going to be found with hair-like projections on top of their cells, on the apical portion of their cells. And so you're going to look at, when you get this portion of the respiratory tract, you look at it under the microscope, this is what you're likely to see. This is what you're likely to see. You're going to see a number of cells there. And if you look at them properly, you will see that these cells are actually pseudostratified. They are not simple columnar. They are not stratified, but they are pseudostratified. So if you look at this one, this is what you're going to see under a microscope. And then you get this and you enlarge it even further, and you're going to see that it is going to be looking like this under that microscope. And then the one other thing you're going to notice is, one, there are these tiny projections on the apical surface. These are known as cilia. The other thing you're going to notice is that this epithelium is going to have some cells that are not going to stain at all. They're not going to stain with whatever stain that is used for you to view this organ under a microscope. And these ones, they are simply not staining because they contain mucus, okay? Because they contain mucus, and therefore these are the goblet cells. These are the goblet cells with their mucus being secreted there and all these. The other thing you will notice is that this epithelium, which is found in the chest, in the air passages of the chest, is going to have pseudostratified columnar epithelium, meaning you will notice that all these cells are going to be resting under a basement membrane. However, even if they rest under a basement membrane, some of them will still have the nuclei at different levels. The normal for simple columnar epithelium is that nuclei is supposed to be at the same level towards the base of the cell. However, in pseudostratified columnar epithelium, the nuclei are going to be at different levels because these cells are closely packed, one, and two, because of the presence of these goblet cells among them. So you'll find that other cells will be so closely packed that the nucleus is going to be moved towards the apical surface of the cell. So the surface epithelium here, this surface epithelium here is cilia. This surface modification of this cell is the cilia, and one of the functions of cilia is for movement of particles, whatever particles are there. So for example, in the respiratory tract, it will be used to move the mucus along the respiratory tract. Along the respiratory tract, these cilia will be moving back and forth in order to sweep the mucus from the respiratory tract, and it will come to your throat, and you're going to clear your throat, and then you're going to spit it out, or you're going to swallow. Okay, so those are some of this specialized surface modifications. The other specialized surface modification that I would like us to look at is keratin. Okay, the other surface modification that I would like us to look at is keratin. So you're going to find keratin on stratified squamous epithelium. Some stratified squamous epithelium will have keratin. So for example, when you are looking at the skin is one of the stratified squamous epithelium. It's one of the organs made up of stratified squamous epithelial tissue. So when you look at the skin under a microscope, this is what you're going to see. And then when you get this skin and you magnify it, this is what you are going to see yet again. All right. When you magnify it even further, this is what you're going to see. Remember, I said that when it's stratified epithelium, the cells may be of different shapes. So you'll find that at the bottom there'll be different shapes of cells, and at the top there'll be different shapes. But the stratified squamous epithelium is named according to the type of cell that is on top. So you're going to find that on top here, if you have squamous epithelium, then even if at the bottom there is cuboidal or whatever, you are going to still call it stratified squamous epithelium. Now, when it comes to epithelial tissue that has keratin, for example, the skin itself, then you are going to call this epithelium keratinized stratified squamous epithelium. Keratinized stratified squamous epithelial tissue. Okay, keratinized squamous keratinized stratified squamous epithelial tissue. So this epithelial tissue type is found in skin, and even areas such as the outer part of the anus. Okay, so some areas of the mouth, you're going to find that they have keratin on them. Keratin is specialized in protecting the skin. Keratin is simply dead skin cells from the bottom of this layer of the skin. The cells are growing to the top, and as they grow, they fill up with this protein known as keratin. And this keratin will then, these dead cells that are filled completely with keratin will then line themselves on top here. And when you bath, the top layers will come off and more will continue coming up like that. So one of the functions of keratin is protection. It protects us from abrasion, 1, 2, even from losing too much water when sweating, as well as even absorbing water when you go swimming or when you're showering. You may end up absorbing water, but keratin protects us. Now, we have looked at the cell shapes, classification of epithelium according to cell shapes. We have looked at classification of epithelium according to the surface modifications. We have looked at the classification of the cilia according to okay, so I was saying that we have looked at classification of epithelia according, 1, to cell shape, 2, according to the number of layers of the cells on this type of epithelium, and 3, according to surface modifications of these epithelia. And so therefore we can safely say that we can clearly name the epithelia now. So one example of epithelial tissue that I'll give you is keratinized stratified squamous epithelium. This is squamous epithelium that has keratin on top of it and has several layers, and it is normally found in the skin. The second example I will give you is pseudostratified epithelium. This is epithelium, normally pseudostratified, by the way, is ciliated. So you're going to find pseudostratified columnar ciliated epithelium, and this type of epithelium is normally found in the air passages, as well as organs such as the fallopian tubes, where the cilia will be helping to move the ovum along the tubule. Okay. The other example that I'm going to give you would be simple cuboidal epithelium. simple cuboid epithelium is usually found in areas where there is secretion. For example, the sweat glands, most glands actually will have simple cuboidal epithelium. And then you're going to find simple squamous epithelium, which is going to be found in places of excretion. For example, the kidneys. You're going to find squamous epithelium in such organs, simple squamous epithelium in such organs. And then you're going to find non-keratinized stratified squamous epithelium. Non-keratinized stratified squamous epithelium is found in places where there's a lot of abrasion, where there is need for protection. For example, the mouth, for example, the vagina, where these organs which undergo a lot of abrasion and they need protection. So you're going to find that stratified squamous epithelium, non-keratinized, is the one that will be found in such areas. Then we have simple columnar epithelium. Simple columnar epithelium is found in areas where mostly there is need for absorption, all right, where there is need for secretion, okay? So organs such as the stomach, organs such as the small intestines, such as the large intestines, that is where you're going to find simple columnar epithelium. Now, we are done with the classifications of epithelia. Now we're going to look at the functions of epithelial tissue. We've already talked about secretion. That epithelial tissue which is specialized for secretion. For example, simple columnar epithelium found in the GIT. Absorption. Some epithelial tissue is specialized for absorption, also found in organs such as the GIT. All right, and this is also simple columnar epithelium. For absorption, we may also find some pseudostratified columnar epithelium found in the epididymis that is also specialized for absorption. Transportation, where some of the cells may have cilia on top, and these cilia allow for movement of particles along the tubule, whatever organ it is. For example, like I said earlier, the fallopian tube. Protection, for example, the skin, okay, and receptor functions, okay. So the epithelial tissue is also functions in receiving and transducing external stimuli, okay. These include issues like the tongue. The tongue has epithelial tissue which is specialized for tasting, such as taste buds. There's olfactory epithelium in the nose, which is specialized for smell. We have in the retina, we have epithelium specialized for sight. Even in the ears, we have epithelium that is specialized for hearing, okay? Okay. So like this is what we were saying, there's simple or pseudostratified, simple squamous epithelia, stratification of epithelia, all these things we have already talked about, okay? Just go ahead and read them. When you look at a diagram like this, you should be able to see and to recognize what type of epithelia this is. When you look at it, you should be able to recognize one by the shape of the nuclei on top of that layer of epithelial tissue, okay? All these are pictures showing you how the epithelium is going to look. So this is a drawing of simple cuboidal epithelial tissue, but this is an actual micrograph as seen under a light microscope. And so this is the lumen, and this looks like a lumen of a gland, of a gland, of a duct of a gland. All right, so you may be able to recognize these things the way they're looking. So under a light microscope, you may not see properly like this, but you will be able to tell. When you look at the cells, the nuclei, the shape of the nuclei, you'll be able to tell that this is cuboidal epithelium just by the shape of the nucleus that you're seeing under the light microscope. Okay, so like this, you will be able to see that this is cuboidal epithelium. Oh, sorry, this is columnar epithelium, because look, the nuclei are towards the basement, okay, membrane, towards the basement membrane. And so you'll be able to see that this is columnar epithelium. Also, this one, you can be able to see that it is columnar, but because the nuclei are at such different levels, this is likely to be pseudostratified columnar epithelium, okay? And so this is how you're going to be identifying all these epithelial, all these epithelial tissue. Okay. Okay. So now, internal structure of the villi, internal structure of the microvilli. Okay. Microvilli, those microvilli, the specifications, the modifications that are found on top of absorptive cells in the intestines. These structures contain a core of 20 to 30 actin filaments that are cross-linked by several actin bundling proteins. So there is one, you find proteins such as valine, okay? We'll find proteins such as fascin, espin, and fibrin. So they are all arranged in an intricate manner such that they are going to be allowing for these stereocilia to work the way they're supposed to work in the absorption of nutrients, okay? So it has valine there is espin, all those to my tiny, tiny proteins, okay? So that is the structure of. Now we also have other proteins such as myosin. Anyway, all these tiny, tiny proteins, you may want to study them and know which ones are which and which ones are interconnecting what. All right. So that is the structure of a microvilli. Now, the structure of cilia. Cilia is made up of microtubules. Remember the microtubules that we talked about in the cell? So these microtubules are arranged in a 9+2 kind of arrangement. I think I mentioned yesterday when we were doing the cell, a 9×2 kind of arrangement. Sorry, a 9+2 kind of arrangement where you have 9 pairs of microtubules. So like 1, 2, 3, 4, 5, 6, 7, 8. This is a cross section. So for example, this is the longitudinal section. When it is cut like this and looked at on top, this is what you're going to see. You're going to see 9 pairs of microtubules and 2 central ones. So this is what we mean by 9+2 pairs of 9+2 pairs of microtubules. And so that is how the cilia is made inside. Cilia is made the same way as flagella, like the flagella of the spermatozoa. This is how you're going to find it. However, okay, anyway, so that is how you are going to, that is what you're going to see once you look at the cilia from inside. Okay, Sometimes you will find some cells that will have only one cilia on top. Instead of a lot of cilia, there will only be one cilia on top, and that is known as the monocilia. And we have monocilia on a lot of eukaryotic cells. Okay, we have a lot of cilia on a lot of, we have monocilia on a lot of eukaryotic cells. And for monocilia, they'll only have 9 pairs of microtubules, and they will not have the central 2 pairs of these microtubules. Monocilia are found in epithelial cells of the rectal testis, in the male reproductive system. That is a part of the tubules of the testis inside. And then we are going to find monocilia in epithelial cells lining the biliary tract. The biliary tract of you remember the gallbladder, okay, that biliary tract there. So you're going to find monocilia. This is an example of monocilia. This is an apical surface of a cell, one cell, and it will have only one cilia on top like that. And the other micrograph is also showing us this monocilia coming all the way from this cell and projecting up, and that is only one cilia. This cell is also showing us one cilia. This is just one cilia, but then it is moving. At this point it was at this position, then it was at this position, then it was at this position. It is moving, and so this is why you're seeing that it is moving from one place to another, and that is also monocilia. Now, one of the major functions of monocilia is to sense. It's for sensation of flow. That is why you're going to find it in places where you're going to find fluid. For example, in the kidneys. For example, in the ependymal cells of the nervous system. Those ependymal cells that are specialized in formation of cerebrospinal fluid. So you may find some monocilia there that senses the pressure and the movement of that fluid. And then the next type of cilia that you're going to find will be nodocilia. Nodocilia is usually found in the embryonic disc. As there is development of the embryo, you will be able to understand this once we start doing embryology. Okay. All right. Now, the other parts that you would be interested in knowing would be the lateral domain. The lateral domain of the cells. Remember, we have the top part, we have the bottom part, but then we also have the sides of these cells. Remember, we said that they are closely opposed, meaning they are held together. So if it's people, they would be held together in such a way that they form a mat. A lot of people held together, meaning towards the front, you're connected to somebody, towards the back, you're connected to somebody, towards the right, you're connected to somebody, towards the left, you're connected to somebody, and at the bottom on your feet, you're connected to the basement membrane. So that is how this epithelium is made. So now in the sides, on the lateral domain of the cells, we are going to have several things, and some of the things that we're going to have would be adhesions. Okay, the lateral domain is characterized by the presence of unique proteins called cell adhesion molecules. Okay, some of these adhesion, the adhesion molecules are they are grouped in different groups. So we have occluding junctions, then we have adherent junctions. All right, so under occluding junctions, we have one, the zonular occludens. The zonular occludens are impermeable. They only allow epithelial cells to function as a barrier. They allow some things to pass through, but very few of those things. So if you look at this diagram, we are looking at this part. These are occluding, like you can hear the word occluding. Now, this blue part here is one cell that has been opened at the back in order for you to see what is happening inside. This is another cell. This is another cell. The only difference is that these two cells have not been opened, but this one has been opened in order for you to see what is happening inside. So if you see this part here, we find that this cell here is closely connected to this cell, which is appearing blue, by these junctions known as tight junctions. The other name for tight junctions is occluding junctions. These junctions are found, they are the ones that are found at the topmost part near the apical portion of the cell. And one of the functions of those occluding junctions is to prevent the passage of just about anything. There will be very few things that will pass through those junctions. So they are also known as the zonular occludens, occluding junctions, or tight junctions. So it is an intercellular diffusion barrier, so that things will not just be passing from one area to another. They prevent the migration of lipids and specialized membrane proteins between the apical and the lateral surfaces, thereby maintaining, okay? So now, thereby maintaining the integrity of these two domains. So when you look at these cells, if these, we are saying that these are the tight junctions, the zonular occludens, the occluding junctions. It means they will not allow things just to pass anyhow. They do allow very few substances, very thin substances, and those thin substances may pass, but that is those are very tiny substances. And then when they allow those things, those tiny things to pass, that is known as paracellular pathway. However, some of the things may pass from the apical side, meaning the luminal portion of the cell, on the luminal side, all the way to the other side, below the basement membrane, below the lamina propria, and they may pass within the cell. And this type of passage is known as the transcellular passage of substances. And so those are some of the ways in which substances move across epithelial tissue. Okay, so we have transcellular and paracellular pathways. Okay, and then we have anchoring junctions. We had first occluding junctions, under which we have talked about the zonular occludens, also known as the tight junctions. All right, now we have anchoring junctions. Remember, we are talking about specialization or modifications of the lateral sides of the cells. And so we are saying that the lateral side of the cells will have certain junctions that one either hold the cells tight to one another, or hold these cells, or allow things to pass through, or disallow things to pass through, or hold these cells to the basement membrane. So now we are saying that those same junctions are grouped. The first group that we looked at is the occluding junctions. And the occluding junctions, we have looked at one which are known as the tight junctions or the zonular occludens. The zonular occludens are the most superficial. They are towards the apical surface of the cells, and they hold the cells together. Okay, and then we have anchoring junctions. Okay, these provide mechanical stability to epithelial cells by linking the cytoskeleton of one cell to another. Remember, when we were looking at the cell, I told you that the cytoskeleton will be found close to the close to the cell membrane, okay, to the cell membrane. So those cells, the cytoskeletons will be held together by these anchoring junctions. So this is, you remember I said this is one cell, this is another cell, this is another cell, and you have cytoskeleton right there under the cell membrane. This cytoskeleton will be linked to the cytoskeleton of another cell membrane by these anchoring junctions. Okay, so there are two types of anchoring cell-to-cell junctions, and these include one, the zonular adherens. Okay, they include one, the zonular adherens. These zonular adherens are a continuous band around the cell. So it is like a belt around the cell, and that is why they are known as the zonular adherens. Okay, with at that same zone. So it will anchor on this side to another cell, on the front it will anchor the cell to another cell, on this other side it will anchor the cell to another cell. These zonular adherents are composed of proteins, different types of proteins, some of which are E-cadherin, okay? Some of which are E-cadherin, okay? So that is how you're going to see. So you've seen these are cytoskeletons here, and they're being anchored. They are forming this anchor that connects them to the other cell. This is one cell, this is another cell, this is an intercellular space here in the middle. But these two cells have been held together. The second type of anchoring junctions are macular adherents. Macular adherents. These macular adherents are also known as desmosomes. These desmosomes, they are anchoring cell-to-cell junctions that provide a particularly strong attachment. Okay, they also interact with intermediate filaments. They create and maintain the structural unity of the epithelium. So these are the desmosomes. All right, these are the desmosomes. The first ones that we looked at were the zonular adherents, and these are the zonular adherents which look like the belt, like there's a belt around the cell. And then we have these desmosomes. These desmosomes, they are going to have certain proteins that will be running from this cell to, remember we talked about the transmembrane proteins, those that are running from this membrane, from the inside of the cell to the outside of the cell. And so those are going to be attached to the cells, to the membrane proteins of the next cell, and they're going to form this desmosome, okay, which is going to be anchoring one cell to another. All right, so this is what we're going to look at. So remember, we said initially we talked about anchoring junctions, and then these anchoring junctions, we said that they are of different types. The first type is the adherent junction, okay? The zonular adherence, and this is the one that looks like a belt around. And then we have the second one, which is the desmosome. The desmosome will be formed by the presence of membrane proteins, which are going to be those transmembrane proteins that are going to attach to the other transmembrane proteins of the next cell. And therefore, you find that they're going to make these kinds of structures that are going to be known as the desmosomes. Okay, so those are some of the structures that are going to be made. Those are some of the junctions that are. And then the other type of junctions we have are communicating junctions. Okay, remember, we have now the communicating junctions. All right, these are also known as gap junctions or nexuses. So one gap junction is known as a nexus. Okay, these are the only non-cellular structures that permit the direct passage of molecules from one cell to another. They consist of an accumulation of transmembrane channels or pores in a tightly packed array where exchange of ions and regulatory molecules and small metabolites occurs. Okay, so you're going to have tightly packed channels, each formed by two half channels called connexons. So those are, like, for example, this diagram here. We are going to have these transmembrane proteins over here, which are connecting to the transmembrane proteins of the other, and these proteins are known as connexons, and they are going to have a channel, okay, a channel through which particles and molecules can pass through from one cell to the other. Now, this is just a summary of the cell junctions and their structural features. Okay, please take time to study it and understand it. Now, the basal domain has specializations as well. One of the things that you will find in on the basal domain is the basement membrane. The basement membrane, it is located next to the basal domain of the epithelial cells and the underlying connective tissue stroma. So you're going to find that between the connective tissue stroma and the epithelial tissue, you are going to find this basement membrane. The connective tissue stroma, which is found under epithelial tissue, is known as lamina propria, which is very loose connective tissue, areolar tissue in nature. The other thing that you're going to find are cell to extracellular matrix junctions, and these are known as the hemidesmosomes or and focal adhesions. Those are some of those junctions that hold the cells to the basement membrane as well as to the extracellular matrix. Now, the basement membrane has a very unique structure. It is, when you are looking at it under a light microscope, you will think it is just one structure. But when you look at it under an electron microscope, you will find that it is made up of two layers also. So it is 10 to 100 nanometers thick, okay, and made up of fine fibrils, fine filaments, okay. These filaments are made up of different types of proteins. For example, laminins, collagen. The major type of collagen found under the basement membrane, in the basement membrane, is type 4 collagen. So it covers about 50% of all collagen in the basement membrane. Okay, and then other things that you're going to find in the basement membrane include proteoglycans and glycoproteins. All right, now when you look at this basement membrane under a microscope, like I said, you're going to look at it and then you're going to see that it is just one layer under a light microscope. However, when you use an electron microscope, you're going to see that it has two layers. The top layer is known as the basal layer, the one which is closest to the basal domain of the epithelial cells is known as the basal lamina. This basal lamina is further made up of two layers, the lamina lucida and the lamina densa. The lamina lucida will appear transparent and glass-like under an electron microscopy. Okay, now this diagram is just here to show us. It is just showing us one. If you look at this micrograph here, this is a picture taken from the kidney, and it is showing us one glomerulus here, one glomerulus here, and this glomerulus, if you notice, there is this structure which is stained pink. It is stained pink. This is the basement membrane on which the epithelial cells are lying, okay? On which the epithelial cells are lying. Now, you get a tiny portion of that, and they drew this a tiny portion of that, and they looked at it under an electron microscope, and they drew it, and they realized that there are two layers of the basement membrane. The topmost layer being the basal lamina, which looks glass-like, and the bottom one, which is the reticular lamina, which looks like it has fine fibrils. The basal lamina, like we said earlier, is also subdivided into two, the lamina lucida and the lamina densa. Okay, so that is how the basement membrane is. This is just another micrograph, and a light micrograph showing you in fact, this should be an electron micrograph showing you the basal lamina, okay? Right there, the one that is looking as if it is white there, right there. And then under it, we have this one that is stained a bit grayish, which is the reticular lamina, okay? So that is about the basement membrane. Okay. Okay, so these are just special micrographs also that may show you more about. Now, some of the proteins of the basal lamina, like I said, we have type 4 collagen, which makes up most of the collagen in the basement membrane. And this is a light micrograph. This one is also taken from the kidneys. Okay, we have a cell here, a cell of the kidneys here, and it has these projections. All these projections are coming from this one cell. And on the other side, we have, this is a blood vessel. And this blood vessel, remember we said that the epithelium of the blood vessel is known as endothelium. So we have these cells, these are simple squamous epithelial cells that line that are making up the endothelium. Between these two cells, between these cells, the endothelium, and this epithelium of the kidneys, we have a basement membrane. Okay, because remember we said that every epithelial tissue should lie on a basement membrane. So in the kidney, the basement membrane is made in such a way that the endothelium will be lying on this basement membrane, as well as the epithelial tissue of the kidney, of the glomerulus, will also be lying on the same basement membrane, in order to allow for the urine to be filtered from the blood and into the glomerular capsule. Okay, so that is what that basal what that basement membrane is talking about. So we have come to the end of today's lesson. Okay, we have come to the end of today's lesson. These are just clinical correlates, the things that you may relate to the anatomy that you have done today. If you have time, you can still go ahead and read a bit through them. Okay, so however, this is the end of today's lesson. See you next time.